Thanks. The co-host. Yeah, it works. Thanks. What happened? Oh. Great. Okay. Hi, everybody. Sorry, it took a couple minutes. I'm I guess I have to zoom the camera. I'm Francis Jeanson. I'm the head of the Center for Analytics here at OBI. And I'm a big fan of hackathons and datathons. I've participated in a number of myself in the past and still hope to do more in the future. This is the BrainHack summer school or school, which is, I guess, affiliated with the Brain Hackathon. And last fall, or I guess fall, winter, and December, I got a chance to participate and showcase a little bit of what we do here at the Ontario Brain Institute, as well as talk a bit about data that's available. That might interest you just as a resource for future projects. So a little bit about me. I did a PhD in cognitive science and AI at Carleton University, and studied sort of evolutionary adaptive systems, cognitive science and AI as an undergrad at U of T. Went to the UK for my master's, but also worked a lot as a sort of software developer and sort of a little bit of a data engineering, and started working actually with OBI in the early days in 2013 to help set up their brain code platform. And I'll talk to you about that in a minute. And then I actually left to work with some startups, worked with some hospitals, and then came back to OBI because they wanted to launch the Center for Analytics, which aims to sort of build on top of what they've done so far with respect to enabling labs, enabling even companies and startups to really advance their capability of building advanced algorithms using multimodal data, and have an impact, ultimately have an impact on patients' health, people with various brain disorders or brain conditions. So it's really nice to have you all here. There's a few souvenirs from OBI. I'll put it, I guess, on the table here. Little booklets and pens. I'll put it in the middle. So help yourselves, leave some for others. Sorry for the folks online. It's only Mariam, but thanks, Mariam, for joining. And how do I hide this? Oh, I guess it doesn't matter. So yeah, let's talk about a little bit about the. No, it's got to be away from behind this.
I can move this way.
I could just slide it.
Yeah, and then you could.
Oh yeah, there we go. Great, thanks. So yeah, OBI. So OBI was actually started in 2010, with the vision that we actually have a tremendous opportunity here in Ontario to showcase excellence in neuroscience research, and the ability to not only showcase research, but also the ability to sort of transform a little bit the way we do research and really engage more with the patient populations, the communities, as well as companies and startups who are developing really new and exciting technologies so that they can better work together. But the big component that's sort of at the heart of this, and this slide sort of showcases this, is the opportunity to look at neuroscience across brain disorders and brain conditions, which is kind of at the vertical purple columns here, but as well across the various types of modalities and types of data that can help better describe what a brain condition is, or even what maybe even a healthy brain is. And you want to be able to look across things like imaging, clinical measures, right? Clinical data that you can collect either from the hospital or even through different assessments that you would have in a research study. Genomics data, as you know, very important, molecular as well. The importance also of common data elements. So that means that collecting common data points, which we do at OBI across the various brain disorders and across even the age groups. And ultimately also make sure that we collect insightful patient outcomes and the results that come out from whatever intervention or any sort of observational events that we're looking at. So this really sort of painted the picture of what OBI's vision was from the get-go, and essentially is what sort of got OBI started in 2010. And since it established a number of programs that are multi-sites, they essentially are not research studies that take place at one university or one hospital or one local organization. They span multiple organizations that have come together to collaborate on major research projects. And it's also engaging the patient population. It's also engaging the industry and commercial sort of entrepreneurs ecosystem, so that they can work together on testing out new ideas, looking for new maybe biomarkers or signs of how a disorder works, and develop new technologies, educate the population, educate even scientists, and ultimately lead to better care. So these programs, let's see. So this is the updated list actually of the programs as of 2023, with 5 major research programs. So these are the circles around here. We have Canbine at the top. This is a major program looking at major depressive disorders in adults. We have PON, we're looking at neurodevelopmental disorders, primarily in children, but more recently, they're also looking at adult populations. CPNet, looking at cerebral palsy, also historically mostly with children and adolescents, but now also looking into cerebral palsy in the adult population. CALM, of which Aaron is a key member of, is a program that Aaron could probably describe in more depth than me, but is looking at multimorbidity in children and youth.
Mental health issues.
Mental health.
That's the person that
You know?
So yeah, now
Yeah.
multiple mental health conditions.
Okay, so you're not using the word.
We're removing
Multimorbidity?
morbidity.
Okay, good to know. So multiple mental health conditions.
Conditions.
Got it.
Yeah.
Okay, thanks. See, you need to rub shoulders with everybody once in a while to get up to speed. And finally, the Transcendent program, which is looking at concussion in the broad population, also children and adults. And so as you see here, they're all connected to a number of different institutions across Ontario, but also sometimes outside of Ontario, like McGill in Quebec, UBC, or I think there was UBC before. Who else do we have? I mean, we have University of Calgary in Alberta, and in some cases, even partners like the University of Michigan in the US, et cetera. So really pan-Canadian and even to some extent international with these other groups outside. And the big vision is to have all these programs, what we call integrated discovery programs, collect this rich set of data so that they can better collaborate and work together. And so to make that happen, we developed a platform called BrainCode. And it's not just a technical platform that brings the data together, it's also a lot of governance and a lot of a framework around how do we enable institutions to work together so that Aaron, for example, can collaborate with a colleague maybe at McMaster or somebody in Ottawa. So rather than them having to do multi-way agreements, 5 would be essentially 5 times 4 divided by 2 for like all types of agreements. Now you just need the 5 agreements with OBI, and then you can collaborate. So it's really a sort of an effective sort of model to enable that collaboration to happen. And on top of that, BrainCode also facilitates and abides by this vision for open science and making data available for secondary use. So for enabling other researchers and other startups or companies who maybe have a new technology that need to validate against some real world data, they can actually come to BrainCode and make a request for access. And if they meet all the checkboxes and they get ethics approval, et cetera, they can obtain access to these data. So over 30 institutions involved, thousands of BrainCode users now at this point. We have data standardization, federation, data linking, and analyses capabilities on the platform. So a little bit more about BrainCode. So this is an updated chart as of this month with over 28,000 records. 26,000 of them are human records and 1,500 are animal records. Over 13 million clinical data points that span all of these programs. And we also have some programs that have sunsetted as of 2023 in epilepsy, for example, and neurodegenerative diseases, and one in concussion. But we have the new programs like Calm and Transcendent, who've started just last year. And so BrainCode collects all this data, multimodal, as I mentioned, enables data sharing, common data elements. So those are the common set of assessments that we have across the population, which is valuable if you want to compare, for example, the performance or the quality of life of somebody with a neurodevelopmental disorder with a group of patients in the epilepsy cohort, for example. And you can actually start comparing those and seeing, are there similarities or are there differences that are relevant, and that could help us better understand their particular condition. And finally, data linking, and that's another important piece that we've implemented, and that this was actually a very hard nut to crack back in 2013, 2014, when we were deploying BrainCode and getting the platform up and running. With data linking, what we can do is we can store encrypted health card numbers, OHIP numbers. And in Ontario, every time you have an encounter with the health system, your OHIP number is typically used. And there's institutions like the Institute for Clinical and Evaluative Sciences who will collect that and aggregate these data sets from the various hospitals, various clinics, and allow research on those data. But they're very, very careful on how you link this data, and maintaining privacy is of utmost importance. And so what we do with OBI is we actually encrypt this using homomorphic encryption. So we can collect these health card numbers, but we can never see them ourselves. But what we can do is using homomorphic encryption, you can actually match them against the data sets at ICES, so that you know that this participant in maybe a CALM study you could also fetch additional health administrative data or any encounters they had with the health system from ICES and do some really interesting research potentially on the relationship between their medical history and other measures that you're doing in your study, like MRIs or genomics or whatnot. So it's a real data discovery opportunity. Some quick numbers in terms of the samples that we have across the programs so far. Calm and Transcendent aren't here yet because they're just getting up and running, but we'll soon have some numbers on them or from them. So Pond, we have clinical imaging data with over 2,700 participants with clinical variables. There's also imaging, there's also genomics. There's sometimes wearable data as well. We didn't have space to put all that here, but we have this table that just kind of highlights the modalities. CPNet has clinical imaging and molecular with over 320 participants. Uplink has clinical, 66 right now. Canbind, across these different modalities, clinical imaging, molecular with 309. Connect with clinical, over 3,000 participants, and Andri had a breadth of various modalities with 520 participants. And some of these programs were still in the process of curating and making this data available for third parties. But this is what's available right now if you want to BrainCode and request access to data. And a little bit about the Center for Analytics. So this is a new initiative by OBI, which I'm heading. Is aimed at building on top of what we've done so far at OBI, which is to work closely with the research groups, develop capacity in collecting the data and in ensuring we have high quality data. What we want to do is further build the skill sets that enable researchers, data scientists, startups to hone their skills in using neuroscience data specifically, and multimodal data in particular. So how do you actually combine imaging with genomics? How do you combine clinical with imaging and genomics, or maybe a wearables data with EEG and MRI? These are real opportunities. The brain is complex, as we all know, and to better understand the conditions that underlie the various brain disorders, I think the overall international community is really starting to realize we need to leverage multimodal data. And we need to build the skill sets to make use of these multimodal data so we can better get to the root of these disorders and build better better ways of diagnosing them, predicting them, treating them. So skills is a big piece, and we're going to be and we are in the process of developing sort of resources and a strategy around supporting the development of these skill sets, accelerating through supporting projects that require high performance computing or advanced, machine learning type computing, such as GPUs. We're now making that available through the cloud and are essentially supporting these projects so that they can achieve this. Another big piece is around creating the tooling ecosystem. So there are a lot of tools that are developed by various labs, but that are sometimes not disseminated very well or others are not familiar with. So we want to help aggregate that, create a knowledge base of tools that are available, and potentially also stage these as pipelines that are readily available for anybody else to use, where they are very popular and could be used multiple times by different groups. We stand to benefit from this opportunity to sort of harmonize when tools are used by different groups so that they can then compare their data more readily. And finally, translation. That's another big piece, and it's really part of the OBI's mandate and vision to have real world impact. We want to support projects that can have some real world impact through translational outcomes. So things that can actually impact on the bedside, tools or projects that can have an impact on improving clinical trials, and really working towards having it, or even being able to inform, for example, the patient population or community. So we're working towards supporting that as well through analysis, AI validation supports. We have a platform that's already privacy compliant. We want to provide additional regulatory compliance support where it makes sense, and advance also the production of apps. Apps that need to be tested in sort of the real world setting will need additional support, and we want to be able to provide that. So that's the Center for Analytics in a nutshell. And now to the meat of this presentation, which is to talk a little bit about data. But are there any questions first before I move on to data? All good? Okay. So some available data sets. So of course, BrainCode, the platform I was just telling you about from the OBI. So you can reach it at brain-code.ca, but also braincode.ca in one word. You don't have to write all these down. I actually have a link to this at the end, and you can just see all these resources. CONP Data Portal is a big initiative. That's the Canadian Open Neuroscience Platform. It started in 2017 and aimed to index a lot of the repositories that we have around Canada. Open Neuro, very rich repository for neuroimaging data. It's public as well, so a lot of the data are available practically just on demand. Dandy as well, Dandy Archive. A lot more sort of neurophysiological data if you're looking for more of this time series, neurophysiological data like EEGs and other types of electrodes. The ADNI repository, this is probably one of the oldest and most rich repositories that we have in neuroscience at the moment. It's focused on Alzheimer's disease, but it is very rich in the sense that there's been multiple studies running for, I think, over 20 years now on ADNI. The ABCD study, if I remember correctly, this is a neurodevelopmental study with some really rich data as well. Nightingale Open Science has a number of interesting data sets available. The NIH Data Sharing Repository have their own sort of index of various data that you can access. The GAIN repository as well for neurodegeneration is quite rich, and GAIN also indexes other repositories. So a very valuable platform. I've used it actually myself in the past. It's quite neat. So that's just a few to start. We also have lists of neuroscience databases on Wikipedia, if you did not know. So you can check it out. BrainCode is on there, but there are others as well. Open Source Brain, a neat project with some more data available. The Mimic clinical data set, a very popular data set in machine learning with, I think it's primarily sort of electronic health record data or EMR data that you'll find on Mimic, but quite rich and valuable if you're interested in that more clinical data. Lion 5B, a multimodal 5 billion AI model that you can access. I started to find these things. Some of them I haven't used in the past, so with all full disclosure, but I think just by going through over them, they seem pretty valuable and rich. Again, Microsoft Azure has an open data set. Google has a Data Explorer, and the bottom one is the NeoGADS, neurodegen datasets that can also be of interest. And then there's the interesting world of synthetic data and models that exist to generate synthetic data. So important to think about in this world where sometimes, in machine learning, you need to balance out your data sets. I was talking to Alice about this morning. And you want to be able to sometimes generate synthetic versions of a data set, especially if you have less of that particular unbalanced group. And so synthetic data sets, and you can ask Alice, she knows quite a bit about this. And there are other benefits to synthetic data sets. When you don't have direct access to the raw data sets, you can sometimes get access to the synthetic data set. So it removes the barriers of time to obtain access. Sinica, for example, is one of those platforms where you can obtain access to the synthetic data set. ADDI is another one. I don't remember if I had that on the list, but we should add that one. And so you can make your own data. Use these tools. You could potentially generate some synthetic data. Some of them are rule-based, traditional, sort of based on common heuristics and common sort of templates for what the data are, and others are based on machine learning, and typically use something like a autoencoder. So it's something that compresses the data and then expands it again. That helps essentially generate similar data with similar distributions statistically, but different properties when you look at the details. Or they can use GANs, these generative adversarial networks as well. So something to think about. There's also tools in R. I think a lot of these on the left work with Python or maybe other platforms, but if you're more of an R user, then SynthPop and SymPop both seem pretty good. And then some really cool models that I want to share. Just the Hugging Face for health models is a really rich collection of AI health data models and various models to make various classifications or predictions on health data. So a very rich repository on its own. And then we have our own OBI tools catalog that we're curating and increasingly adding more tools to that you can also access online, which would showcase a lot of the tools that our integrated discovery programs that I showed you at the beginning are making use of. So I think that's it for the resources. And so as I promised, and I'll move this bar, but you can access all these resources on this bit.ly link and QR code. So you take a quick shot and you'll find it. It'll be easier. And I think it's in edit mode for everybody. So please don't delete, but do please add or make updates if you realize if there's a dead link. Yeah, it'd be great to see more resources on this list. And so I think that's it for me. Oh, a couple examples of some interesting work that's been done by the programs, especially in the machine learning space. Cambine, they were predicting response to escitalopram using multimodal data. So they're using clinical imaging and molecular data. They had 192 participants in that data set. And they tested 5 different machine learning models and found that when they combined clinical and molecular, they had the best prediction of response to the drug with 73% area under the operating curve. And this was using ElasticNet out of the 5 models. And combining early and late treatment data worked the best, according to the paper. So a pretty cool initial exploration of the value and the benefit you gain from combining data versus just using one modality alone. So clinical alone performed worse, right? Molecular alone performed worse. I think they looked at imaging as well, but it performed worse. Alice, you probably know about this paper, but maybe you have something else you.
There's also another one in the, I think genomic. Like you said, imaging, working on the
Yes.
image is very difficult.
Yeah,
Yeah.
right.
And yeah, you do have to combine, with other modality
Right.
to improve it. Yeah.
Right.
Sure.
Yeah. And then another example. So the previous one was looking at response of a drug, right? So trying to predict how well do people respond, because that's really interesting. If you can predict that some individuals respond better, then they don't necessarily have to go through the standard guidelines of care, which they might have to go through 3 other drugs beforehand. You can just jump directly to the one that's going to best work for them. So that's a huge benefit from a treatment perspective. Pon was looking at trying to understand how groups, whether the neurodevelopmental disorders like autism, spectrum disorder, obsessive compulsive disorder, and ADHD, how do they compare? And are those standard clinical labels correct, or should they actually be, re-engineered, so to speak? We need to think about this differently and more in a data-driven way? So what they've done is they actually just mixed all of those participants and looked at two modalities. You have cortical thickness and phenotypic information. 226 participants, and used vague multi-view clustering. This is work by Azadeh Kushki, who's the PI on this, or the lead investigator on this, who's really a super talented data scientist working at Hull and Bloorview. And they did some clustering that showed that there were clusters that formed where individuals with traditional labels would actually be together in one cluster, and other individuals also with those labels might be in another cluster. So the clusters almost reveal in a data-driven way that those traditional clinical labels don't always quite apply well. And so there may be an opportunity for us to start rethinking about how do we actually categorize or classify individuals with these neurodevelopmental disorders. So some really, really neat work, and they're continuing to explore this with even more rich data. So other sort of areas to think about, clinical trials optimization. The drug development fails because of the heterogeneity of participants. So it's important to think about how can we subtype them, classify them, so that we can potentially design better trials that are more effective and more targeted. We want to predict outcomes, and being able to generate these brain fingerprints, for example, can be a way for us to look at how the trajectory of that disorder evolves over time. Comorbidity disambiguation, also an important area where you want to understand the factors that relate to a condition that might also relate to another condition, the comorbid conditions. So, we want to disentangle this web, so to speak, of complexity and various for the various patients and individuals who are each and every one to some extent fairly unique. And so we want to move towards more of that, what we call precision sort of medicine, precision sort of understanding. So many other sort of ideas and concepts to explore from a research perspective, but I think these are just some I want to highlight. And general fields of research, I think important areas to think about: how do we prevent health conditions? How do we detect health conditions? Screening, diagnosis, etc. How do you predict health conditions and progression? How do you treat ultimately health conditions or prevent? If you can, that's even probably better. And how efficient are we at handling these health conditions? And last but not least, how good are the data and tools that we have to do all of the above? And that's an important area of research onto itself. So it's not just about research in sort of the brain disorders itself, but it's also research into the informatics tools, into the way we process data and develop better tools. That's also important. And so you'll hear about the term neuroinformatics. That's one area, but you have neuroanalytics as a common sort of theme. Is how do we actually build the tools and the capacity to do all this great analysis? So yes, that's it. And thank you for your time. Any questions?
Any job opening?
Any job opening? Not right at the moment, but possibly soon. But projects, so with the Center for Analytics, we are launching some projects. So we have the two first projects that were just started. One is looking at the simulation of cortical activity and to predict how do we actually, can we use essentially electrical stimulation to reduce the onset of epileptic seizures, so it's non-invasive. And right now there is no good method that's non-invasive. It's typically invasive where you have to perform surgery. So this group is looking at it's Lyle Mueller and his lab at Western, sorry. We're looking at a way to simulate this cortical activity and using data to confirm whether their methods are working or not. And then another group is Cove Neuroscience, and they have a general rich platform for actually looking at things a bit like you were telling me earlier, Alice, but looking at graphs and transforming neuroimaging data into graph data to understand connectivity and yes, yeah.
So like GNN, you try to like do graph transformations.
Yes, I believe I don't know the ins and outs of the details of what they're doing, but they're essentially building pipelines and platforms to work with the different modalities, like including EEG, MEG, MRI, and others to.
I wanted to work in that area.
Right.
Yeah.
Well, yeah, we can always
like
make intros, and you're happy to connect with them. But this is led by Nardine Samuel at Cove Neuroscience, and she has and Irene and Leaf. We actually have they're doing a webinar next week, so I could share the link maybe with Aaron, and you can, or Juchi, and you can share it with the group. They'll be chatting about their methods and techniques for this graph modeling of brain activity. So yeah, could be interesting. Yeah. Any other questions?
Great presentation. Thank you very much. Two questions. Is there any charge for the companies who want to get access to that?
No, not right now. No, there's no charge. Yeah.
And then how open are you
to independent researchers? I think it can be achieved. Typically, there needs to be sort of an affiliation that we look for, just so that we can better have a sense of the trustworthiness of the individual. But we also combine that with, so we look at sort of the profile, the CV. So the CV of the individual is important. And we also look at, I mean, there's an ethics approval that needs to be obtained for access to the data. And when I say like we call these controlled data sets, this is typically human-based data. There are actually animal data and also phantom scans that you do in an MRI that are available in a public way. So you just simply register on the platform and you can download those right away. So there's mouse model data that you can download. There's this phantom data that you can download right away. But the control data are typically the human-based, from participants in research studies. Yeah, good question. Anything else?
Can I ask what the topic of your PhD was?
Oh, sure, yeah. I
took cognitive science at Carleton, so I'm just
Oh, no
trying
way.
to see the bridge
Oh, cool.
between,
Yeah.
how you.
I was cross between the cognitive science department and the computer science department.
Okay.
I did cortical. I was looking at spatial temporal coding, and we modeled essentially what you call spatial temporal codes where coincidence detection takes place versus rate coding. So typically in the standard sort of theory about how the brain codes information is through frequency, like the firing rate between the afferent and the target neuron. But if you actually look at a population of cells, they can also start to fire in patterns where the action potential, the signal arrives at the target cell almost at the same time, and you can sort of encode information that way and by using the population like temporal dynamics. So that's what I looked at, and we simulated robots navigating a maze and like storing memories essentially in those networks. Yeah.
I have not met another grad
Oh,
I have student
oh, no way.
Carleton yet, so.
Cool, cool.
Exciting.
Did you do your undergrad or
No,
grad?
my master's.
Your master's? Oh, cool.
I
Oh,
did
great.
my master's in science
Oh, nice. At Carleton or U of T?
in Dublin.
In Dublin. Oh, nice.
I think Carleton's might be still the only Canadian one, so, yeah, if I meet someone, it's somebody from overseas.
It's the only grad, I think, one.
Yeah, the only
yeah,
grad.
Yeah,
one,
yeah.
I did the undergrad at York. and then went
Okay.
to-
I did U of T and then master's in Sussex. Yeah. That's cool. Great. Yeah. Some familiar,
Thank you.
some colleagues. Yeah. Thank you, everybody.
Thanks for the list too.
Yeah, no problem. Bye, Miriam.